Hello, AP Biology students. We're going to go over the 10th section in Unit 3. And before we get into the 10th section on light-dependent reactions, I'm going to warn you, there's a lot of information here. Part of the reason why this unit is so difficult is because there's so much to memorize. But I like to remind students that if you kind of make it like a story, it becomes easier to intake and to memorize and to learn about. Before we begin, let's just review where we're at. Remember that the light-dependent reactions are going to require light and water, and they're going to produce ATP and NADPH. These two molecules are going to power the Calvin cycle, which is going to make our main product, glucose. The byproduct here of oxygen is actually going to be made during the light-dependent reactions. All right, so we're going to just dive deeper into what is actually going on in these light-dependent reactions. Remember that we're in the thylakoid or the thylakoid membrane within the chloroplast of a cell. I'm going to go over each step individually in the light-dependent reactions. First, we're starting with water splitting at something called photosystem 2. Whenever you see PS, that's just going to stand for photosystem. Yes, I know it's weird that we're starting with photosystem 2 instead of photosystem 1, but they were named kind of backwards, and now we just keep them the way that they were identified. So again, water is going to split, and you can see that occurring down here. It's going to split into oxygen and hydrogen ions. What it's going to do is actually going to donate electrons to this photosystem. The oxygen atoms that are going to break away here are actually going to form the oxygen that we breathe in. This is the byproduct of the light-dependent reactions. We're going to talk about the importance of these hydrogen ions later. If you're confused about what a hydrogen ion is, remember that a hydrogen atom basically just has a proton and an electron. And when it becomes an ion, it's going to lose that electron. So it's just going to be basically a proton. Photosystem 2 is going to absorb light by using pigments within the photosystem. It's going to transfer that energy into a location called the reaction center. Electrons that are located in that reaction center are going to become energized and move down something called the electron transport chain. The electron transport chain are just a bunch of membrane proteins. They're going to be found between photosystem 2 and photosystem 1. As the electrons move down this electron transport chain, or ETC, the membrane proteins involved here are going to use some of the energy that the electrons have. What these membrane proteins in the ETC are going to do is they're going to further pump hydrogen ions into this space, which is known as the lumen. So now we already have two locations where we're increasing the amount of hydrogen ions in the lumen. We have where the water is being broken, and we have during the electron transport chain when they're funneling more hydrogen ions into the lumen here. The electrons are then going to make their way to the photosystem 1. Here, the electrons are going to become re-energized by more photons of light being absorbed by the pigments in the photosystem. So again, the electrons are going to become energized at this point once again. The electrons are going to pass to another membrane protein called NADP reductase. This enzyme is going to bind NADP, a hydrogen ion, and the electrons all together to form something called NADPH. This is our first product of the light-dependent reactions that's going to funnel its way into the light-independent reactions. You can see that occurring over here where we have NADP and a hydrogen ion and two electrons being bonded together to form NADPH. Now remember that we're in the thylakoids. We're in those photosynthetic discs within chloroplast. And what we've been doing is creating a high concentration of hydrogen ions, or a reservoir of hydrogen ions inside the lumen here. That's honestly one of the main goals of this reaction, is to create that concentration gradient where we have a high amount of hydrogen ions and a low amount of hydrogen ions outside the membrane. What's going to happen is the hydrogen ions are going to move through another enzyme called ATP synthase. ATP synthase obviously makes ATP. And it does this by turning and using that flow of hydrogen ions from the lumen to the stroma. And it's going to turn in a circular fashion and bind a phosphate group onto ADP, making it ATP. This is our other main goal or product of the light-dependent reaction, this ATP. This is a high-energy molecule that the light-independent reactions are going to use. So again, these hydrogen ions are going to move through this ATP synthase, and this enzyme is going to act like a dam. It's going to use the flow of the hydrogen ions to make ATP. We can again see this process of the movement of hydrogen ions from high concentrations in the lumen to low concentrations in the stroma through ATP synthase. This is again going to act like a dam and use that movement using the flow of the hydrogen ions to make ATP. I'm going to quickly go over again the process from beginning to start. Water is going to break down here at photosystem 2, donating its electrons. These are going to become energized by the photosystem absorbing light and transferring it to the electrons, which make them high energy electrons. These electrons are going to move down the electron transport chain, which uses that energy to force more hydrogen ions into the thylakoid's lumen. At photosystem 1, the electrons become re-energized by light being absorbed by pigments and transferring the energy into the electrons. And these electrons are then going to be dumped off onto an enzyme called NADP reductase. NADP reductase is going to bind the hydrogen ions and electrons onto NADP, making it NADPH. This is our first product of the light-dependent reactions. Now that we have a high concentration of hydrogen ions inside the lumen compared with a stroma, they're going to start moving through the ATP synthase, which makes ATP. ATP is our second main product of the light-dependent reactions. They're going to go on and fuel the light-independent reactions. Okay, I know there was a lot that went on there, but our main goal here of the light-dependent reactions is to make ATP, to make NADPH.